try to give a short educational teaser at the beginning, where we try to cover some of the methods that I used later in the presentations. Exactly. And today I would like to cover just one specific aspect that is not only important for the two applications that we will hear later on, but that's generally, I think, a very important point to know when it comes to quantitative proteomics, and that's the difference between relative and absolute quantitative proteomics. What do they mean? When do you need what? And how do you generate such data? Okay, so on the first slide here, I would like to define once more together with you what I mean when I speak about relative protein quantification or absolute protein quantification. Exactly the same, by the way, holds true for metabolomic quantification as well. So when I speak about relative quantification, the important hallmarks to keep in mind is that relative quantification is always performed with at least 2 samples, potentially much more samples, and the aim, the output that we always want to generate in a relative quantitative measurement is a protein or peptide ratio. So we would like to tell at the end of the day whether protein X is 10-fold upregulated or 2-fold downregulated or not at all regulated between the 2 or several samples we're looking at. So what we do in relative quantification is we always compare the same peptide or the same protein between samples. So here you have a representation of the typical outcome of proteomics. We always love to demonstrate our data in form of these heat maps, where all the lines in this table or in these heat maps represent the different proteins that we detected and quantified, and all the columns represent different samples that we analyzed. And then you see the measured protein intensity for each protein in each sample in each of the cells, right? And when we do relative quantification, we analyze such a quantitative table in a way that we always just focus on the lines. So we always just compare the measured mass spectrometric intensity of one given protein across all the samples. And we define usually a reference condition or a reference sample, and we say this protein is up or down or not regulated by X-fold. That's what's relative quantification. How does absolute quantification compare to this now? Now, absolute quantification you can also perform with just a single sample. You do not need several samples. You can, of course, also do absolute quant on many samples, but also just one sample is sufficient. And the one important thing to understand about absolute quantification is that in absolute quant, you can, of course, also compare the absolute concentration of one protein over various samples, but you can do one more thing. You can also compare the concentration of different proteins within one sample. So you can go column-wise and start comparing proteins to each other. And the desired output that you want to generate at the end of the day by absolute quant, most people consider as a real protein or peptide concentration. So at the end of the day, you would like to say whether protein X or protein Y has a concentration of a certain femtomole per microgram of tissue or nanograms per milliliter of blood, or whether protein X is at a certain copy number in a cell. These are all the typical absolute quant units people like to work with. And this is what most people consider absolute quant. And I always call it, it's the prime way of doing absolute quantification, or the Rolls-Royce approach. But what many people do not consider so much as absolute quant is that actually not necessarily an output of an absolute quantification is always a concentration, but also a protein or peptide ratio can be the output of an absolute quant measurement. But then this is not the ratio of the same protein between samples, but a ratio of different proteins within one sample. Also, a ratio can be an absolute quantitative measurement. And then, yeah, you would say whether protein X is X-fold more abundant than protein Y, or protein X represents a certain percentage of the total protein content in your sample. Okay, so the challenge to do relative versus absolute quantification is not the same. Actually, doing accurate relative quantification is more easy than doing accurate absolute quantification, because there is a number of issues that are much more influencing absolute than relative quant. So for example, if you don't manage to completely lyse your cell, maybe a relative quantification is still accurate, but absolute quantification can be very wrong. Also, if you have issues during sample preparation in terms of extraction efficiency, digestion efficiency, all of these are things that are much more important or much more troublesome if you do absolute quant. And then there is also this issue of signal intensity that we see in our mass specs. And I will come back to that in a minute, which is also a really serious issue for absolute quant. Okay, so what the important thing to keep in mind in proteomics, we are very good in doing the relative quant. We are very good in just focusing on one analyte, whether it's a protein, a peptide, or a metabolite, and to then say, how does it relative change over samples? And this is the type of analysis that Klaus, for example, or that we used in Klaus' project, where we looked at novel proteins that have so far not been described, and where we followed the expression of that protein over various samples and show that it's regulated over a growth rate. But then there are also some projects where absolute quant really matters. And what's important to keep in mind is that absolute quantification is typically less accurate than relative because of all these challenges I told you. But it's still sometimes worth and important to tackle these challenges and do absolute quant. And we did that in Julia's project, because here we really wanted to be able to tell how pure a purified protein sample is. So we wanted to compare concentrations of different proteins in one sample to each other. And that's why we tackled absolute quant in her project. Okay, and on the next slides now, I just would like to give you a brief idea of the different methods we have in proteomics to achieve absolute protein quantification. And I already mentioned that one challenge that really impacts absolute quant is the issue of mass spectrometric signal intensity, which is not necessarily only correlated to concentration. And this is something I want to demonstrate you here based on an example. So let's assume we measure this protein here with proteomics. So what we usually do is we digest it with an enzyme into its peptides, and then we measure the peptides in the mass spec. And the signals that we will see for the different peptides in the mass spec will be vastly different. Okay, there we will have very high intense peptides and very low intense, even though in theory, all of these peptides have the same concentration because they originate from the same amount of protein. The reason why this issue occurs in proteomics is that these intensities that we see here depend, unfortunately, on a lot of more factors than just the concentration of the peptide in the sample. So from a simplified point of view, concentration is one factor that determines the intensity, but then we have, for example, also digestion efficiency. So some peptides are just not excised from the protein as complete as others. We have recovery during sample prep. Some peptides are very hydrophobic. We tend to lose them during the sample prep because they attach to vessel walls. Ionization efficiency of peptides, just some peptides ionize much better than other sequences, matrix effects, all of these combined factors together influence the intensity that we see then later on in the mass spectrometer. And the proteomics people always call this the MS response factor. So each peptide has an own unique response factor, and that determines the concentration. Now, for absolute quant, this means that if we just measure the intensity of a peptide in a sample and we want to infer the concentration, this is very difficult, right? If we don't know this response factor, the intensity says nothing about the concentration. All the other things need to be taken into account as well. And this is why, in order to do absolute quant, you need to determine the response factor of a peptide. Only then you can perform this transfer from intensity to concentration. Now, there are two general ways how you can learn the response factor. The one way is to use isotope labeled reference standards. So to do absolute quantification and learn MS response factors by working with isotope labeled reference peptides. So for each peptide or each protein you want to absolutely quantify, you need a reference molecule. And in proteomics, we work a lot with synthetic peptides as reference molecules, but you can also work with completely labeled proteins as reference molecules. And by measuring then these reference molecules in form of a dilution series, so what you usually do is you take your sample of interest and you then spike into your sample various varying concentrations of this pure reference, isotopically labeled reference molecule, and thereby you determine this response factor of this molecule, and then you can very nicely quantify the concentration of the endogenous, the light peptide in the sample. This is the standard way and the most precise and accurate way, if it's done optimally, to do absolute quantification. But downside, this is a very time and also money expensive workflow, because these isotope labeled reference molecules are very expensive, and you really need, for every analyte, for every peptide or protein, you need the matching reference. So it is nothing that you can do at a large scale. You always just do it for very few proteins. And in the light of these shortcomings, people since many years already thought about alternative ways how you can also estimate absolute quantification, omitting any of these isotope label standards. And this is called absolute label-free abundance estimation in proteomics. There are various different algorithms published that try to do exactly this. And what all of these algorithms have in common is all of them somehow try to normalize for these different peptide intensities that we see here. Okay, so what the algorithms try to do is to get rid of these differences that we see. And they use different ideas in physics how they normalize the response factor. And here you just see 6 examples. There are even more out there in the field. And the most popular one, or the one that is used most frequently, is called this IBAC algorithm. It's not because it's the best algorithm, but it's implemented in a software. It's called MaxQuant, which is used by a lot of proteomics people. So it's very easy to have it. And this is one major reason why a lot of people like to work with it. And the basic idea of IBAC is just that in order to estimate the absolute concentration, what you do is you sum up all the peptides that you could quantify for a given protein. So in this example, you would sum up all the intensities you've measured here. And then you divide this sum of all peptide intensities by the number of theoretical peptides a protein can produce. So, which means you just normalize by protein size pretty much, because you take into account larger proteins produce much more peptides than smaller ones. And the basic assumption of IBAC is that this distribution of peptide intensities that we see here for a given protein is very similar for all the proteins that exist. So every protein produces some good high responder peptides and some low abundant peptides. And by summing them all up, the distributions are very similar, and the response factor is constant. This is the underlying assumption of IBAC. A very easy algorithm, very easy theory. And people have spent a lot of studies have tried to see how good that works. This is one paper from 2013 where they tested various different absolute label-free quantification approaches, and they worked in this sample with a gold standard sample. So they worked with a commercial sample where you have 50 human proteins in defined concentration mixed together. So you know the absolute concentration of these proteins. And they tested these different algorithms, and what they found is that the absolute concentration you determine with these algorithms, it's in the range of 1.5-fold wrong concentration compared to the correct one. So the average fold error was 1.5-fold, and 90% of the proteins had an error that was smaller than 2-fold. So is that good? Is that bad? This is the type of range you can achieve using these absolute label-free quantification methods. Exactly, and we use this approach now in Julia's project, IBAC, because Julia was interested, or she's working, as she will tell you in her talk, she's working with purified proteins from SARS-CoV-2 for a microarray experiment. And her question was, how pure, how good are these protein samples that I have to put on my microarray? And we use the IBAC algorithm to generate plots like this, where we can see for each sample that she uses, how pure is this, has this protein purification, overexpression and purification, how good was it? And then we had some samples that looked very good. So 98% of the peptide intensities that we found in this sample are all coming from the target protein, so the NCAP protein in that case. And some other samples look maybe less good. So here only 88% are the target protein, and we also found some significant contaminants there. So this is the type of data that you can generate with this IPAC algorithm and doing absolute quant. Okay, I think that's all I wanted to say. So the general pros and cons of this label-based and label-free is really that the label-based approaches for abs quant are more precise, but they're expensive and limited to a few targets. And label-free is really an algorithm if the absolute accuracy is maybe a bit less important, but if you want to quantify also lots of molecules and if you don't want to invest a lot of money into standards. Summary, what I think would be great if you keep that in mind. So proteomics data will always provide you large tables with proteins identified and samples that you quantify these proteins. And what you do in relative quant is you always stay in one line. And the type of questions that you can ask then is this protein 1, how is it regulated over the different samples? And you find this protein is low in sample 1, 2, 3 and high in sample 4, 5, 6. So maybe 1, 2, 3 are biological replicates of a diseased person, and 4, 5, 6 are replicates of a healthy person, and then this is a very interesting biomarker for you. And proteomics is very good in doing this type of relative comparison. But proteomics can also do absolute quantification and thereby compare different proteins in one sample. And then the plot that you generate is per sample, and now you have all the proteins listed here. And here you will find out now that protein 4 is the highest abundant protein in that sample, and it's twice as abundant as protein 5 and 10 times more abundant than protein 2. And to do this type of absolute comparison, you either need isotopically labeled standards or you need to work with a specific algorithm for absolute quant, and IPAC is one of them. There are many others as well. So this is our educational introduction into the methods the people used. Are there any questions from the audience about?
So thanks for the opportunity to present here. And of course, thanks a lot for the MS measurements that have been done of my samples. And I very much hope that my talk will be interesting for all of you, although it's not actually about MS, but rather about bioanalytics. So first of all, let's talk about SARS-CoV-2, although you probably don't want to hear about it anymore. So here you can see the virus with its main genes and its main structural proteins. And if somebody is infected, his body will form antibodies to all of these structural proteins. While if someone gets a vaccination, all of the currently applied vaccinations only use the spike protein. So people with a vaccination will only have antibodies to the spike protein. So that's just a short, important remark at the beginning about the antibody response to SARS-CoV-2. And now if we take a look at the current infection numbers, we can see that although we've been in this for more than 2 years now, it's still not at the end. We've had more than 350 million cases so far. This is approximately 4.4% of the world population, which is really a lot. 5.6 million people have already died from an infection with SARS-CoV-2. And luckily, we are now able to vaccinate. So the first vaccines have been approved like a bit more than a year ago. And 9.9 billion vaccinations have been done since then. But still, there are a lot of open questions that still have to be answered. Like, for example, to what extent are antibodies formed after infection or after vaccination? Is immunity after infection comparable to that after vaccination? Which one is better? How long does this immunity last? When do we need booster vaccinations? And as you all know, we now have the Omicron variant. Are the vaccines also efficient for the mutated virus versions, or don't they help us at all with them? And one tool that is very helpful in answering these questions are immunoassays. Immunoassays that are able to detect antibodies to certain proteins from SARS-CoV-2. And so we decided to also develop such an immunoassay. And what we do is chemiluminescence microarray immunoassays. So we have a microarray chip reader, it's this analysis device, and we use microarray chips that can be made either from glass or from polycarbonate. And on these microarray chips, we have antigens immobilized. So in the case of SARS-CoV-2, we mainly use the receptor binding domain, the S1 protein and the N protein. And these are immobilized on the surface of a chip. And then we simply inject a blood sample into the microarray chip. And the antibodies that are contained within that blood sample can then bind to the antigens on the surface. And subsequently, we add a detection antibody that will also bind to the bound antibodies here. And then we can do a detection using hydrogen peroxide and luminol. So we get a chemiluminescence reaction here. And this reaction is recorded using a CCD camera. So in the end, we get a nice image. So all these bright spots that you can see here are spots where antibodies have bound. And with this principle, we need really low sample volumes. We need about 8 microliters of serum or plasma in a 1:5 dilution. And what is the most important thing about our test, many of the typical commercial tests are ELISA assays that take some hours to be carried out. And our test has a very short sample interaction time. Therefore, the whole measurement duration is under 4 minutes. So you can really wait for the result. You just take a blood sample, inject it into the device, and then you have your result in under 4 minutes. But of course, we need antigens that we can immobilize on the chip. And these are produced by a company that is located in Planegg. It's called ISA Bioscience. And the main proteins we got from them were the M protein of SARS-CoV-2 with 421 amino acids, the receptor binding domain with 223 amino acids, and they also produce some other proteins that are already applied in preliminary tests. So some mutant SARS-CoV-2 RBDs, the S proteins from some endemic coronaviruses, and the SARS-CoV-2 S2 protein. And these proteins are all produced in Chinese hamster ovary cells or CHO cells. This is very important as it's a mammalian expression system, which also allows for post-translational modifications, making them very similar to what these proteins would look like in the human body. But of course, this also might give rise to impurities, to hamster proteins that are still left within these target proteins that we actually want to use. So we were very happy that we got the MS measurements from Tina and that she could tell us if we still had to do some work in the purification or if the proteins were looking good. And now if we have our proteins, we have to immobilize them on the microarray chips. So if we use glass chips, it's just basic microscope slides. These are silanized and subsequently surface modified using Jeffamine. Jeffamine looks like this, so it has terminal amino groups. And these are then immobilized on the surface, and the proteins can bind to these amino groups via either their carboxyl or amine groups. So it's very basic protein coupling chemistry. We can also use different coupling strategies depending on whether we want to use the carboxyl or the amine groups. And we bring the protein onto the chip by contact printing, which is basically very simple. It's a needle that transfers protein onto the chip. The drawback of these glass chips is that there's a very high work and time expenditure in producing them. So it takes 66 hours or 4 working days to produce one batch of chips. And therefore we were looking for faster production methods. And what we found was polycarbonate chips. Polycarbonate is a very suitable base material because it's cheap and it's very easy to handle. So we just use polycarbonate plates and cut them in smaller parts. And we then again apply a polymer, which is in this case a modified Jeffamine. So it has a carboxylic group at one end. It's applied by screen printing, subsequently incubated at elevated temperature. And then basically we do the same as for the glass chips. So again, we use microcontact printing of our proteins, in this case with EDC sulfur NHS coupling chemistry. So again, very basic protein coupling chemistry. And then the only thing we still have to do is to assemble the microarray chip. So it's the spotted chip. And this is a double-sided adhesive foil with a flow cell in it. And then we can immobilize up to 100 protein spots on this chip and can easily measure it. This already sounds good from the idea, but does it actually work? So we tested it with 65 pre-classified samples with both glass and polycarbonate chips. And we then detected cutoff values by ROC curve analysis and did a background correction and normalization with respect to these cutoff values. And this is the results we got. I'm only showing the positive samples here. You can see a dotted black line down here. It's very small. That's the cutoff value. So as you can see, all of the samples were found above this cutoff value and therefore classified correctly. Same is true for the negative samples as well. And on top, you can see the polycarbonate chips. And on the bottom, you can see the glass chips, which had been measured on the previous device generation. So the results are very similar for all of the tested proteins, which are N-RBD and S1 protein. And with both chip types, we could classify all samples correctly. So in general, the principle seemed to work. And we then thought about possible applications because we wanted to go further than that. So we found some different applications that might be useful in the future for serodiagnostics, not only in SARS-CoV-2, but also with other infections, because I still hope that this pandemic at some point will be over and we can then use our technique for other diseases as well. So the first thing I want to show you is the whole blood measurements. Because if we want to do a point of care test that is rapid, that gives you the result directly after you inject the sample, then we should be able to use blood from a finger prick. So we don't have to draw blood. We don't have to do any extensive sample preparation like centrifugation of the blood. And therefore, we simply tested if we could use whole blood. We therefore used 60 microliters per measurement instead of 8 microliters, which were necessary when using plasma or serum. This is due to the hematocrit effect. So in the whole blood, we still have cells. So a whole lot of the volume is basically cells. And we then compared plasma and whole blood measurements from a total of 21 donors and found that the background signal does not increase upon use of whole blood. And we still got the same diagnostic sensitivity and specificity. So we still classified all of the samples the same way, no matter which sample type we used. So for the future, we can use blood from a finger prick without any problems. The second thing we focused on was the distinction of convalescence and vaccination, because that's something many commercial SARS-CoV-2 antibody tests can't do. They only determine antibodies to one protein, and if that is the S protein, they cannot distinguish if the antibodies were formed after vaccination or in an infection, because in both cases, you will have antibodies to the S protein. And on the other hand, there is tests that only use the N protein. So these might not detect vaccinated people at all because they don't have antibodies to the M protein. And here, a big advantage of our microarray can be used. So we can immobilize various proteins and can then distinguish convalescent and vaccinated samples based on the signal pattern. As you can see here, negative samples will only give bright spots in the positive control row, while a vaccinated sample will give spots for the receptor binding domain of SARS-CoV-2 and the S1 protein of SARS-CoV-2, so the parts or the proteins that are contained within the vaccines. And if we have a convalescent sample, we also get a signal for the M protein. So this can also be very helpful if one wants to detect vaccination breakthrough infections, because these might go unnoticed. And if we do these measurements after vaccination, and if we do them regularly, we can maybe detect these infections and get more information about that. And if we go a bit further on that, we can also do serosurveillance after vaccination. So this is relevant to find out about the necessity and the timing of booster vaccinations, because now we are already talking about the next booster vaccination, vaccination number 4. And to see how the antibody titer would change after vaccination, we measured a total of 12 samples from the same person that had been taken between 268 days before and 117 days after the first Pfizer-BioNTech dose. Apart from the receptor binding domain, we also monitored the M protein, as I just explained, because we wanted to check for possible breakthrough infections. And this is what we found. So as you can see here, for a very long time, until 7 days after the first vaccination, both the RBD and the N signal are below the cutoff that is shown in black here. And then 15 days after the first dose, the receptor binding domain signal goes up, and it reaches its maximum 14 days after the second dose. So this is in concordance with what Pfizer-BioNTech saw in their studies. And then 14 days after the second dose, the decline starts. And as you can see, it's really fast. So we lose antibody relatively fast. And therefore, we can use these measurements to check if a booster vaccination might be helpful or if it's not yet necessary. And another thing we can see here is that the N protein stays below the cutoff at all times. So no breakthrough infection occurred. And then the last thing I wanted to show is the sequential detection of IgM and IgG antibodies. Because usually in an infection, first of all, we have a high pathogen load. So this is the phase where somebody really is infected. And then the antibody production starts. And first of all, the body produces IgM antibodies that will then again fade relatively fast. And afterwards, IgG antibodies are formed. So if we are able to measure both of these antibody types, we will get information on the time course of an infection. To measure both antibody types, we simply executed 2 assay cycles within one measurement. So in the first measurement, we only detect the IgM because all antibodies will bind to the antigens, but we use a specific detection antibody for IgM antibodies and therefore only get the IgM signal. And then in the second step, an IgG detection antibody is added, and we can detect the combined signal of IgG and IgM, and therefore we can simply calculate the IgG signal afterwards. We tested this with 24 samples, and as you can see here, the samples that were negative for IgM and IgG showed values below the cutoff in all measurements, so in the IgM and the IgG measurements. The samples that were positive for both showed values above the cutoff in all of the measurements. And the samples that were positive for IgG only show values below the cutoff for the IgM measurement and then above the cutoff for the subsequent IgG measurement. So all samples were classified correctly. This is not very helpful with SARS-CoV-2 because here the IgM production starts relatively fast. That's also why we don't have any samples that only have IgM but no IgG. But it will hopefully be helpful in the future for diseases where IgM is really formed very early during an infection. And with that, I'm already at the end of my talk. I just want to give a short outlook on what we want to do in the upcoming months, or more like the upcoming years. So we are planning to do on-chip neutralization assays because neutralizing antibodies are even more important than the total antibody count. We want to do T-cell measurements on our chip. And we are currently already working on the measurements of antibodies to different SARS-CoV-2 mutants. So here Tina again helped us by providing MS measurements of these mutants, and we still have to do a bit of purification there. And then last but not least, we are planning to extend the antigen panel to endemic coronaviruses, influenza, and in the future possibly to a general vaccination status control. And with that, I would like to thank everybody who works with me in that project, which is by now quite a lot of people. And thank you all for your kind attention.
Thanks for the invitation, and thanks for listening. It was very interesting hearing more about SARS-CoV, even though yes, we're kind of fed up with this virus. However, we switch gears and let's talk about Pseudomonas. So Pseudomonas aeruginosa is a common bug which is present all over the place. It's kind of a dirt organism, sometimes living in water, but it's everywhere found in the environment. We work with strain PA01. It has a genome of 6.2 megabase pairs, about 5,500 open reading frames, and a relatively high GC content, so 66% roundabout. E. coli, just to remind you, has about 50%. Okay, and Pseudomonas aeruginosa has its name from the green tint it produces, so that's a mixture of pyocyan blue and pyoverdin in green, and the name aeruginosa, I have just read in Wikipedia, translates to verdigris or copper rust. That's the greenish tint you get on copper if it is outside on a roof, say, or this green you have for the Statue of Liberty, this green color. It's copper rust. So Pseudomonas is named after that. Pseudomonas aeruginosa is able to invade many hosts. Actually, no species is safe from this bug. Plants, then you have biofilms. I haven't found nice pictures for insects, but I have found two pictures from humans, which I only show briefly because they are a little bit yucky. So you have Pseudomonas, for example, sometimes below the nail or in the eye, and you see the green tint. Okay. So our main focus actually are novel genes, and we were interested in these very peculiar type of genes, so-called overlapping genes. When we started this project, we applied a technique which is called ribosome footprinting. This technique is a sequencing technique, and this should be able to tell us on whether a gene is used. So you produce a messenger RNA, and then you have the ribosomes on top of it. Here in red, the messenger RNA, and then the ribosomes in blue. You isolate those translating ribosomes from a bacterial strain of your interest, Pseudomonas, E. coli, whatever you like. And then you apply an RNAs, and the RNAs will eat away the messenger RNA between the ribosomes. So you get the mRNA fragments within the ribosome. You open the ribosome, so you just isolate the messenger RNA fragments, and these are called footprints. And because they have been in the ribosome, you know that these pieces have been used to produce a protein. Otherwise, they wouldn't have been in the ribosome, right? Okay. Maybe I get a better color here. Yeah, that's good. So when we now have the genome of a strain, so you see the open reading frames, which are known to produce messenger RNA at first and then a protein later. We can measure along the gene these footprints of the translated messenger RNA, and these are these little reads which you can see here. And this gene has a relatively weak value, so low number of reads, and here high number of reads for this gene. That you have these gaps and valleys and hills, this is well, due to the technique, it's not perfect, it's not covering everything, and actually, to be very honest, we don't know why you sometimes get stacks of reads like this one here, and sometimes you don't get any read at all. It has to do something with the speed of the ribosome running over the mRNA, but there are other effects we don't understand yet. But nevertheless, we see nicely reads on top of this gene. Okay, so we can tell this part of the genome has been translated. It was first an mRNA, now stuck in the ribosomes. Very good. Okay, how to go forward? This way. Okay, so with ribosome footprinting, we now detected unusual transcription translation patterns. So this is the transcriptome, the standard RNA sequencing. Here you sequence the mRNA. Okay, and when you do that, you see for some genes high coverage with mRNA. Certainly you have to do it strand-specific. That's, I haven't said, but you know, it's strand-specific. And here for this gene, funny enough, you see reads also on this side of the genome, right? So there is some RNA produced, and normally people would assume this is non-coding RNA, which may regulate the actual gene messenger RNA. So normally people assume non-coding RNA if it is not within a known open reading frame, right? Okay, then we took our ribosome profiling. So here the question, non-coding RNA, or is it maybe messenger RNA? So we took the ribosome profiling, and suddenly we see we get also ribosomal footprints. So the messenger RNA had stuck in ribosomes and was translated. Not strongly, okay, this is strong, relatively weak, but still we get a nice signal. And if we do an overlay, okay, translated mRNA, we see this for a gap, so to speak. So here's a gap, and there fits nicely a long open reading frame. And the funny thing about this now with the overlay is that we here have an overlapping gene. So we have an open reading frame, which is highly transcribed and translated, as you can see. But we also have another gene, which is transcribed and translated at the same time. So these are overlapping genes. Okay, so in Pseudomonas, we found two of these unusual genes, OLG1 and OLG2. So the names are overlapping gene 1, overlapping gene 2. Quite fancy. And thing is that we use Pseudomonas aeruginosa because it has a high GC content, as I told you in the beginning. And high GC content means long open reading frames. This has to do with the stop codons. The stop codons are TA rich. So in a high GC, you get longer ORFs. This is why we used Pseudomonas. Okay, here is the standard experiment. RNA sequencing for all genes now in this violin plot. And our genes we're interested in, OLG1 in blue and OLG2 in red, are shown here for the standard RNA sequencing experiment, right? And it is just as the bulk of all other proteins in the middle of the diagram. Okay, and as you already know, because the title already told you, we're talking about overlapping genes. So the common overlapping genes, the common annotated genes which overlap are these two here, and they're also in the middle of the bulk. So then we did ribosome footprinting. So looking only at the fragments sticking in the ribosome, and you can see same. Here's a picture for all genes, a violin plot, and these are just within the bulk. Then what we also can look at is the number of ribosomes sitting on top and how many parts of the messenger RNA are covered with the ribosomes. And you see this fits nicely with the most other genes we see. And what we also can see is how well the messenger RNA is translated. So some are translated extremely good, but only very few, and most are translated around 1-ish. So this is a standard kind of factor. Okay, so let's proceed. Okay, so we found this unusual open reading frame in the genome of Pseudomonas to be transcribed and translated. Here we have the gene arrangement. So this is the overlapping gene 1. This is the mother gene, so to speak, the gene where overlapping gene 1 overlaps with. And here are the values, the reads in a little bit different type of picture. Here you can see the reads for the ribosome profiling, here for the standard messenger RNA profiling, so transcription. Transcription and translation. Okay, so same for OLG2. Here, mother gene, the known annotated gene, and here's a novel open reading frame for which we also found transcription and translation. All right, so far so good. But, well, overlapping. That's now the big question mark with our colleagues. This is either very, very great or fake science, right? And what we persistently get from our colleagues is that overlapping genes are viewed as some oddity. Meanwhile, this has changed, but only after about 15 years of research in the group of Siegfried Scherer at first and now in my group. So we have over the years published several of those genes. So this was the example I've shown you, published 2 years ago, and there are some other examples published some years ago. So we're getting there, right? But still, people would doubt if overlapping genes exist, because the ultimate proof for microbiologists is to show the peptide, right? Mass spectrometry, and this is why we are here. Okay, so we turn to the BayBioMS, yay. And as you all would have expected, I mean, that's why we're here, for the overlap, for the mother gene, for the common, normal, annotated, known gene, we see, okay, there are peptides, yes, in mass spectrometry. Very good. So we picked a few, shown with the asterisks, and added some label peptide to the reaction, as Tina has explained in the beginning. So we bought for these 5, with the asterisks, we bought label peptides. So we, yeah, we put the money out of the pocket. So, and it worked for 4, which are black, and for 1 it didn't. And interestingly enough, we also found peptides for the overlapping gene 1. And we confirmed using heavy peptides 4 of those. Excellent. Yes, that was really a breakthrough. This was also for us the first time that we could show that, yeah, we have mass spectrometry data. So the protein is produced, and here we really have proof of it. This is quite unusual. So, and as I said, the asterisks, the peptides were used for parallel reaction monitoring. So we ordered the heavy peptides, and some were validated and quantified. This is the filled asterisk and the empty asterisk. There it didn't work. So we could not verify this peptide found here, but never mind, we found 4 other ones, and here we found all 4 for this overlapping gene. Cool. Okay, same for overlapping gene 2. You've seen this picture already. So a lot of peptides for the annotated gene. Some were picked for PRM. And here, overlapping gene 2, we also picked. And yeah, well, it wasn't as good as we expected, but at least 1 peptide we could verify, right? Okay. Alrighty. So we now took samples from a growth curve, and here you see for overlapping gene 1 after 1 hour, after OD1, which is around about here-ish, and 24 hours, we measured the peptides using this PRM. So we had these heavy labeled peptides, and we measured the amount, and there you can see here's a scale of the annotated gene and the within embedded overlapping gene, and we see a nice time curve. So it's starting to be produced after 1 hour. Peak is at OD1, and at 24 hours you couldn't see anything, neither the annotated nor the overlapping gene. It was different for OLK2. Remember, we only had 1 peptide for OLK2, which you could do the measurements. Nevertheless, 1 hour it starts, OD1 peak, 24 hours it's gone. In contrast, the annotated gene is produced and stays. Okay, so here is a genetic loci. So we did promoter analysis, looked at where the transcriptional stop is, and so on, and looked at the whole locus. And here are the annotated genes on the genome, and here's this novel open reading frame where we now have confirmed not only by messenger RNA sequencing and ribosome profiling, but also on top mass spectrometry that this thing is producing a protein. and this also. So both genes on both sides of the DNA strand produce protein. Same for this one, promoter analysis, terminator here, and so on. Okay. And these open reading frames are in frame -1, just for your interest. So that means that the triplets are the same, but just read in the other direction, right? In the one or the other direction, but the same triplets. Okay, so then we did some analysis concerning evolution. This was actually done by Zachary. He's also here in the show. And we could see that this overlapping gene 1 is restricted to a few strains. Overlapping gene 2 has a little bit of broader distribution, and down here are similar open reading frames, but they have stop codons. Hard to see, but all the black bars here are stop codons, so they're probably non-functional. So the undisturbed open reading frame is only present in a few strains here and a little bit more strains here. Okay, then we looked for evolution, and we found negative selection or purifying selection or removal of alleles that are deleterious. So the genes seem to be used in the bacteria, but only in a few strains, not in many. So these are all the other strains, and well, you have some signals here, but basically the open reading frame was absent for all the other strains. Okay, so finally, we have more overlapping genes found in the past. I would say that we have about, yeah, it's hard to tell. So when we started this project, we were very enthusiastic, and I think still I would say about 10% more genes in the bacterial genome can be found. They are overlapping genes. However, evidence is hard to come by if you talk about mass spectrometry. Nevertheless, for Pseudomonas, we have found some more with peptides. Some of them are unfortunately so-called one-hit wonders, means you have only one peptide, and one peptide in mass spectrometry is for most of the people nothing. But we still have the data from ribosome profiling, so I think we can publish a few more. And we have similar results, but without mass spectrometry for EHEC. And this is in a preprint and is sitting there quite for a while, trying to get this published, but the colleagues are a little bit hesitant. Overlapping genes, so many, do they exist? Have they a function? Is this really true? And so on and so on. So this is not so easy. So I would like just to acknowledge for this talk, Michaela Kreitmeier. She was first author of the publication. Zachary did the evolutionary analysis. Siegfried Scherer was the chair. He is retired last spring. Then we have the Bavarian Center for Biomolecular Mass Spectrometry. Yes, since they showed for the first time overlapping genes in mass spec. So this was work of Miriam with the help of Christina. And then finally, we get money from the German Research Foundation and also internal funds. If you have questions, you can email me, and thanks for being here.